So the good news, now we're moving away from that chemistry section in there to more stuff on cells. Yeah, you still can't see it with the naked eye, what's going on, but it's at least not chemistry for you. So in this section we look at cell communication. In here, and this is going to be both how the cells communicate with other cells, but also how they communicate inside the cell, what is going on in their external environment. So the external signals need to be converted into responses within the cell. And one of the important things here is cells can identify mates by chemical signaling in there. And we don't always think of cells mating, but in a sense a lot of them do. So when we talk about them being transduced, it's going to be the cell surface that is going to be changed into a form that brings about the cellular response of mating. And it's not mating like we think of with sperm and egg in there. It can be a lot of times that they come close together and that genetic information can be swapped between them. Bacteria are notorious for doing that. That's how genes for resistance get passed from one bacteria to another, is they get close to each other in there and they're able to actually make a copy of that gene and pass it on to their buddy in there. So the signal transducing pathway is going to be that you have a signal that's going to attach to the cell surface, and then it's going to go through a series of steps as it's going to be converted into the response inside the cell. And this is going to be very similar in yeasts and mammals, and then plants and bacteria will be more similar in how they handle things. Another thing that can happen is that cells can be quorum sensing. This can allow bacteria to sense the density of other organisms around, and a lot of times they can coordinate their activities in there based on what's going to benefit the whole population in there. So even though they're all individual organisms, they will actually start to work together as a mass doing that. It can also mean that they will look and say, you know what, there's too many other bacteria here, don't divide, we've got enough in the population. That's one of the ways that some of the good bacteria in your body will actually inhibit you from getting an infection. Your gut relies on this a lot. Is a new bacteria will come in there and think, okay, I'm going to come in and divide and do all these things, and it gets in there and it says, this is too crowded, I'm not going to do this here. It can actually sense how many other bacteria are around it. They can form biofilms, which is what happens when you have these aggregations of the bacteria, and they will a lot of times take on specialized functions. So one of the places in you where you're going to have biofilms is on your teeth in there. That they will actually take on their own function as a mass together in there. So these signals can be both local and long distance in there. So it can be local with the immediate neighbor right next door, or it can be things that are a little bit farther away. Both animal and plant cells have these junctions specifically for this purpose that are going to connect the cytoplasms of adjacent cells. Animal cells can communicate by directly contacting surface molecules and do cell-to-cell recognition in there. And these are all going to be influenced by local regulators that are in the vicinity, helping to decide which of these things are going to happen. So some examples of these, you've got growth factors. In animal cells, these are compounds that are going to stimulate the target cells to grow or divide in there. So with cancers, they're going to be having these growth factors that are going to behave differently. Sometimes they end up creating their own growth factors to promote their own growth. But this is going to be part of when your body knows when it needs to make new cells and when it doesn't need to. With paracrine signaling, numerous cells can receive or respond to growth molecules produced by a single cell at a time. So it doesn't have to always be one cell tells another cell to do this. You can have one cell telling a group of cells you all need to grow in there. And synaptic signaling is going to be in animal nerve cells, where the electric signal is going to trigger a release of a chemical signal called a neurotransmitter that's going to stimulate a target cell. So it's going to send the information from one cell to another. So for longer distance signaling, we use hormones for this. In the body, a hormone is a substance created in one location that acts on another in the body. And so this will happen in both plants and animals. With endocrine signaling, that message is going to travel through the circulatory system. Plant growth regulators are going to travel through vessels, or some of them will diffuse through the air as a gas in there, so plants can actually respond to gases that are around them. An example would be if you've ever taken and put something in a brown paper bag with bananas to ripen it. The bananas just happen to produce a lot of the ethylene gas in there that will act as a signal to tell a plant to ripen. Avocados, if you can find one in this area that's actually ripe, will do that as well. You usually have to use your bananas to ripen your avocados here. And so that's an example of a gas causing that signal into the plants. And then transmission of nerve signals can also be a way of long-distance signaling. So with nerves, it's acting locally, but it can carry it over a longer distance. So you can have a nerve signal that starts in your brain and ends up sending a message down to your hand of what to do in there. So the 3 stages of cell signaling, this is pretty straightforward in there. You've got reception, that's going to detect the signal molecule when it binds the receptor. The transduction, where it's going to convert the signal into a form that carries out a specific response, and then the response that's going to be actually triggered and carried out by the cell. So when we receive these signals in here, it's going to bind to a receptor protein, and a lot of times it's going to change its shape. They're going to call these ligands in there, and a ligand is just a molecule that's going to specifically bind to another, and it often is going to cause the receptor to change shape. I don't know why this keeps wanting to scoot over on the screen. So that's going to act a lot like a lock and key mechanism. This ligand thing is how your antibodies are going to target specific things. There are so many of these ligands out there, we don't have a clue how many are out there really, because they're still finding new ones. You actually have ligands that are going to tell your body this is where we should store fat. So if you've ever wondered why it tends to cluster in some areas on your body and not others, it's because in some areas you've got more of these ligands than others, and it's just going to go there whether you want it to or not. So some of these receptors are going to be located in the plasma membrane, and they'll end up binding to that specific receptor protein, and it's going to transmit the information to inside the cell. And a lot of these are, common one is the G protein kinases. So if you remember, a kinase means it moves phosphorus or phosphates on there. Coupled receptors and tyrosine kinases. So you actually have tyrosine kinases that are involved with making pigment in your skin in there. The intracellular receptors are going to be in the cytoplasm or the nucleus of the target cells. And in these cases, the signal molecule has to be small enough and hydrophobic that it can actually get into the cell. So an example would be nitric oxide, which is going to cause blood vessels to expand, and steroid hormones. A lot of those can end up controlling genes. So sometimes it just bypasses the outside of the cell, it diffuses right in to send its message in there. Steroid hormones, when they're used as medications, will oftentimes have a lot of side effects because they are able to diffuse into any cell like that. So they will act as transcription factors, which will be proteins which are going to control which genes are turned on and off. So your G protein coupled receptors. With these, you've got a plasma membrane receptor that's going to work with the G protein, and it ends up binding GTP to get things started. So some bacteria are actually going to interfere with the G protein receptors, and that's going to be how they will make you sick, is by interfering with some of that cell communication in there. So this is a typo in there. Let's say some. So the G protein is going to be attached on the cytoplasmic side of the membrane, and it's going to function as a molecular on and off switch in there. So the cytoplasmic side, remember, is the inside portion, and it's going to cause the GTP to displace GDP, acting kind of like the ATP does in the ADP, where you're going to have a phosphate come on and off in there, and it will activate a G protein that will dissociate and diffuse along the membrane and binds the enzyme to activate it. So it's basically starting a cascade of events where you attach to one receptor, it's going to activate something else, sends it's on its way to activate something else in there. That's how these work. So changes in the enzyme in G protein are temporary, so they can be reused. So when you want to stop that reaction, the steps basically reverse themselves and they go back to their original state. So another group of these are the receptor tyrosine kinases that are going to have enzyme activity where the phosphates are going to attach to tyrosine. So it's a kinase, it's going to transfer phosphate groups. So your receptor has individual peptides with an extracellular ligand binding site. So there's going to be that ligand or receptor on the outside of the cell. And it's going to have a helix portion that's going to span the membrane. And then there is an intracellular tail on the other side of it that's going to have these kinases in there. So where the other one had to actually interact with something on the inside of the membrane, just on the inside surface, this one spans the membrane in there. The signaling molecule is going to end up binding, causing the polypeptides to closely associate and form a dimer. It's going to change shape in there. And then it activates the tyrosine kinase to add a phosphate from ATP onto the tail of the peptide, and then the receptor is fully activated in there. So it's going to, again, act as a relay protein to bind that phosphorylated tyrosine, changes the structure, and is going to signal transduction pathway for the cellular response in there. So you'll notice with these tyrosine kinases that ATP is involved with it. This is going to be one way that you actually use up some of that energy that you are creating through metabolism in there. If you look at how much energy you actually create and you go, okay, well, I exercise and I burn 500 calories of energy by exercising, but I consume 2,000 calories. Why am I not just growing fatter and fatter and fatter from that? This is how you spend a lot of your other energy in there, is through these communication pathways in the cell. This is where a lot of other ATP gets used up, is in making these communication pathways. So another type is the ion channel receptors. So this time your ligand is going to be specific for an ion in there. So these are called the ligand-gated ion channels. And it's going to act as a gate, and when the receptor changes shape, it opens the gate. So Sodium-Potassium Pumps are going to be an example of one of these. So when you have the sodium bind to it, it changes shape that moves it to the inside. On the inside, when you have potassium bind to it, it changes shapes and moves the potassium to the outside in there. So it's going to bind the ligand, binds the receptor till it closes and it's going to stay closed until it binds the receptor. Then it's going to open the gate, allow the ion to flow through, and then when it dissociates, it's going to go back to its original form in there. Sometimes what's going to decide whether or not you open or close that gate is going to be a change in the voltage. And this is what the nervous system relies on, is a change in the voltage in there to send that message. So if you want to actually stimulate a nerve, you can do that artificially from the outside environment using electrical voltage. That's what electric stim does, is it applies that electric voltage from the outside, and it's causing these channels to open up in there. So some intracellular examples with steroid hormones. Steroid hormones are going to be things like testosterone. It's going to pass through the membrane, and it's going to bind and activate a receptor in the cytoplasm. And then that entire complex is going to enter the nucleus, and it will bind to a specific gene, creating a transcription factor in there. So it's not that these signals just are going to influence what's going on inside the cell. They can actually bind onto the DNA and determine which genes you're going to turn on and off. So if you were to take the hormone testosterone, and females have some of it anyway, but put artificial levels of the testosterone into a female, both sexes are going to have, for the most part, the same genes in there, and very few of the genes are on the Y chromosome. So you can have them be activated by testosterone. So when you put that testosterone in a female, it's going to trigger some of the same things that go on in a male. So it isn't that the Y chromosome has a gene in there that says, let's make males have thicker skin and coarser body hair. That's a gene that's in everybody. So if you put that testosterone in a female like that, you can turn those genes on and turn yourself into a thick-skinned, hairy woman if you really wanted to in there. So we're at halfway here. I'm going to go ahead and pause it. Let's go ahead and continue on. So when we have transduction in a cell, it's just a cascade of interactions that relay the information from the outside into the inside. And many of these are going to have multiple steps. Most of them have multiple steps. But by doing this, by having multiple steps like that, each step is a potential place to amplify the message. And that's why it's set up that way, so that you can try and coordinate and regulate what's going on downstream from it. So your signal transduction pathways, they are going to bind a signaling molecule in the plasma membrane. And then that receptor signals a chain of molecular interactions that lead to a response. If that original signal molecule trigger is not passed along, it usually never enters the cell. So that can be at one point where you can end up regulating multiple pathways in there at the same time. So say you need 5 different enzymes to digest something, rather than having to turn off 5 different enzymes individually. If you have them all regulated by the same signal, you only have to turn it off at one point in there. So it's kind of like having multiple lights on one switch. I can turn off a whole bunch of lights all at one time with one switch that way in a classroom. So did you have thought I heard a question coming. Okay. With protein phosphorylation and dephosphorylation, a protein kinase is going to transfer phosphate groups from ATP to the protein. So that enzyme kinase, again, just means transferring phosphates on there. Most are going to act on proteins different from themselves, unlike the tyrosine kinase example. So that's going to act on tyrosine, but with the other examples, it can act on other proteins. So a lot of these relays in a signal transduction pathway are protein kinases in there. And they're going to be in a phosphorylation cascade. So it means you're going to move the phosphate from one substance to the next, to the next, to the next in there. A lot of these are going to regulate cell reproduction, and when you have abnormal regulation of these pathways in there, you can contribute to cancer. So as I mentioned, there is the tyrosine kinase pathway that's involved with pigment of the skin. And one of the ways that they will try and control pigment of the skin with aging things is they will use tyrosine kinase inhibitors in there to stop those pigment spots that will occur on the skin. So if somebody's had a lot of sun damage where they will get a lot of freckles, they'll put these products on there to try and stop more freckles from forming from the pigments in there. So protein phosphatases are going to be enzymes that rapidly remove phosphate groups. They're going to do dephosphorylation. And these are going to be mechanisms for turning off the signal and making protein kinases available for reuse. So all of these enzymes and pathways in there, we don't want them to be consumed in the process. We want them to be able to be reused in the process. So the phosphorylation cascade example, you have your relay molecule activate a protein kinase, and it's going to transfer a phosphate from ATP to the protein kinase to activate it. That active kinase 2 is going to catalyze phosphorylation or activation of a protein kinase 3 down the line, and that kinase 3 is going to phosphorylate a protein for the cell's response. So it's just passing it down a chain of events in there. So your small molecules and ions as second messenger pathways. These you're going to have a small non-protein water-soluble molecular ion that's going to act in the signaling pathways. So these are going to be a lot smaller. Your protein kinases, tyrosine kinases are fairly large. This is going to do a similar way of regulating, just using something small in there. And because they're small and water soluble, these messages can be spread out more by diffusion. So the reason you would choose to use this over one of the larger ones is you can spread this out in a body a lot faster in there. So your cyclic CAMP or your cyclic AMP is adenosine monophosphate in there, is a really common one in there. And you've got your adenyl cyclase, that's the enzyme in the plasma membrane, and it's going to convert ATP to CAMP in there in response to an extracellular signal. So this is an example with epinephrine in there. Epinephrine is what's going to be released in your body when you're in the fight or flight response in there. It's going to bind that receptor protein, and it activates the adenylate cyclase, and then it's going to synthesize the CAMP, and it can make a lot of those to amplify the message. And then the phosphodiesterase converts CAMP to AMP to end signaling. So it's again having one signal come in, and it's going to pass the message to something else in there. It acts almost like a translator of what's going on outside. So the CAMP is a common one in there. When you look, a lot of medications are going to alter this CAMP pathway in there. It's a way that you can stop a communication going on to create a desired effect in the body. So the CAMP is usually going to activate another protein, and protein kinase A is going to be one of the examples. It's going to be sometimes referred to as a serine or a threonine kinase in there. And the inhibitory G protein in there can inhibit this pathway, and it can be activated by different signaling molecules. So another type of signaling pathway we use are the calcium ions in there. And these are going to be based on the concentrations of calcium in the cytosol. These are used a lot in animal cells, and it's going to act as a second messenger system, meaning it's going to have a cascade of events that triggers a response in something else. So your cytosolic calcium is usually going to rise by a mechanism that's going to release it from the endoplasmic reticulum. So in a muscle cell, this is what's going to happen. When you trigger and send the message to a muscle cell that you want it to contract, the nerve's going to send that message there, and there's going to be a cascade of events that go on in the muscle cell membrane that will end up triggering the endoplasmic reticulum in the muscle cell, which we call a sarcoplasmic reticulum, to release calcium, which allows the muscle to contract. And so you've got your iodine triphosphate and diacylglycerol messengers that are going to be cleaved using certain phospholipids in the plasma membrane in there. So with these, that's going to be part of the regulation in there. You've got your signal molecule that's going to come in and bind the receptor. It's going to activate your enzyme phospholipase C. It's going to cleave the phospholipid in a plasma membrane in there to make your diacylglycerol in there and your IP3. That's going to act as a second messenger, triggering other pathways in there. So this ends up going to control those calcium ion channels in the endoplasmic reticulum, allowing the calcium to flow out into the cytosol, raising that gradient in there, and then it's going to activate the next protein in the pathway. So in the muscles, what it does is it ends up binding to what covers your muscles to keep them from contracting. So it's just the next step in the cascade of events. So the responses that can happen as a result of this, so you can have responses be in the nucleus or the cytoplasm of the cell in there. It can regulate the synthesis of a protein or the activation of a protein in an activity in there. Some of these will actually lead to cell division in there. So these are going to help to regulate everything that goes on in the cell. It can help to fine-tune a response in there, so it can help to coordinate a response going on with something else in the cell. It can, with the amplification, you can turn a response up or down, so the amount and intensity of the response can be controlled in there. So each catalytic step provides another potential place for regulating things in there. So the specificity of these, different cells are going to have different proteins in there. You can have the same trigger different proteins in there. They're still all going to have to fit specifically with it. It's not going to be random in there. But that is one way that you can have one signal go out and control multiple things in there, is multiple proteins could hold that trigger in there. So an example would be with epinephrine. If you release this in your body, it's not going to target just one area in your body. It's going to be able to target your liver to release glycogen. It's going to fit another protein on your heart to speed up your heart rate. It's going to fit another protein in the muscles in your eyes to cause your pupils to dilate. So that's an example of having one trigger spread to multiple places in the body like that. So another thing that we run into are scaffolding proteins, which are large relay proteins to which other relay proteins are simultaneously attached. It's another way of making this more efficient because it's going to help the enzymes find their mate. If they all go to the scaffolding protein, they know where to find things. It's kind of like saying this is a singles bar for all of these to go. They know that's where you're going to go look to find your mate rather than randomly floating around all over the place in there. So the scaffolding proteins are permanently going to hold together networks of signaling proteins at synapses in the brain. So there are places in your brain that are going to be kind of like hubs of information where everything is going to go there to find who it needs to communicate with. So to terminate a signal, we need to sometimes be able to keep a cell alert and responding, and we want to be able to have that signal last only a short time. So if you send a message to your muscle to contract, you don't want that to stay permanently. You need to be able to send another message to it to uncontract in order for you to be able to repeat that action in there. If you don't have that signal end with your heart, it would contract once and it would push blood out once, but you wouldn't have that pump continue to go over and over again. So the end of the signaling is important for a lot of reasons like that. So when you bind a signaling molecule, it has to be reversible in there, so that when you take that signaling molecule off, it goes back to the original shape. So it's kind of like being able to go up and down with the light switch in there. When you put the light switch back down, the lights need to go back off in there. So apoptosis, this is kind of an interesting thing with cells. It's really important. A lot of times people would go, well, why would it want to do that? This is a pre-programmed cell death. And cells will tend to look out for the greater good of the organism that they are part of in there. They're not always out just for their own best interest to live as long as possible. The ones that are doing that are cancer cells. They're the ones, they will kind of tend to act in their own best interest, and they've started to ignore what's in the best interest of the organism they're living in there. So you're going to have multiple cell signals integrated in here. So during apoptosis, your cellular agents actually start to chop up the DNA inside and the organelles and the cytoplasmic components, and the cell begins to shrink and become lobed. They call it blebbing. And parts get packaged up in vesicles so that when that cell dies, scavenger cells can come along and engulf it by doing phagocytosis. And this is really important. This goes on a lot in us during embryological development. And so they look at it in the soil worm. It's going to play a crucial role during embryological development. It's been studied well in there. But this process going on in you is what lets you have individual fingers and not webbed fingers. Apoptosis is part of that process in there. You've got capases that are proteases of apoptosis. When the cell receives its death signal, these are going to become activated in there. So in humans, we have, that we know of right now, about 15 different capases that carry out apoptosis in there. And the pathway used is going to depend on the cell and the type of trigger in there. So often the signal is going to originate outside the cell that it's time to die. There's something in the environment that's going to tell the cell that it needs to do that. Sometimes the signal can originate inside the cell in the nucleus. So for example, if your DNA has become so mutated and so messed up, one of the ways that the cell can look out for the greater good of the organism is it says, you know what, I just can't fix this. Let's just wipe it out and start all over. And it can activate a capase for that. And one of the things that will happen is it kind of starts to make one of those proteins, and it makes it, and it's so misfolded and contorted that it says, you know, we can't use it. And that protein that it can't use will actually be what sends that signal in there. So in developing paws and feet in there, so failure to do that can result in webbed fingers and toes in humans. It also, we think that it's got involvement in certain degenerative diseases like Parkinson's and Alzheimer's in there. So just like you don't want to have a failure of apoptosis, you don't want to have apoptosis going on before it's supposed to either, and degenerating and breaking down your tissues too early. So your cell cycle, we'll go ahead and get started on this in here. A cell cycle is going to be its cell life cycle from the time it is first formed until it divides into two cells. So with cell division, it's going to result in two genetically identical daughter cells. So we're talking about non-sex cells here, just your normal body cells or normal individual cells that take one cell and split into two. So when you do that, both of those cells are genetically identical to the parent that they came from in there. So it's going to have a genome in there that is its genetic information. Your chromosomes are going to be that packaged form of the DNA that is going to be used for replication. That's going to be really the only time you're going to see it be in the form of chromosomes is during replication in there. So when we talk about it being a somatic cell, that is a body cell. Soma means body. That is one of your cells that is any place except the sperm or egg. A gamete is a reproductive cell. That's the sperm or the egg in there. They end up having half as many chromosomes as the somatic cells because they fuse with somebody else's gamete and bring half of the chromosomes from the other person in there. So the chromatin is going to be the complex of DNA that is loosely coiled in there. It's going to have its associated proteins with it. So your DNA is going to be arranged in these chromatin fibers when it's not dividing. What it kind of looks like if you see it enlarged enough in a cell is like if you took a ball of string and you just took it and threw the ball and you're holding on to one end of the string and it uncoils and then you sweep it up into a nice pile in there. That is what your chromatin looks like. Completely random coiled up string. You can think of it if you're sitting on the floor maybe doing some homework and you're working on 5 different classes and you've got pieces of paper all over the place, kind of like what my office looks like in there. You've got everything out so you can access it in there. And that's the reason the cell does that, is things need to be nice and loose so you can actually get enzymes in there to look at the DNA. The chromosome is when you coil it all up and put it together to be able to move it and divide it. And that's the only time it does it. Kind of like my office again. The only time it's going to be clean and organized is when I have to move it. So your sister chromatids, when you have your chromosomes, it looks like an X in there. And the two halves of the X are the sister chromatids. You've got one sister, the other sister with it. And they're each going to contain an identical DNA molecule. So you have 2 copies of it in there. You've got cohesins, which are adhesive protein complexes that are going to hold them together. So if we look at it like this, you've got sister 1, sister 2. Your cohesin is going to hold them together in there. So the centromere is going to be the specialized region of the chromosome where the 2 chromatids are closely attached. So your centromere is that region where the cohesin holds it together. The cohesin is like the sticky substance in there. So when you think adhesive, cohesive on there, the sticky substance in the centromere is where it holds. Later on in cell division, those sister chromatids are going to separate and be considered the individual chromosomes. And then each nucleus is going to receive a collection of the chromosomes that are identical to that of the parent. So mitosis is the process of division of the nucleus. We usually look at it in terms of the whole cell dividing. There are some cells that will have the nucleus divide multiple times, that will undergo multiple mitoses, but the whole cell doesn't divide in there. So in cell biology, they will divide it up, and the difference between mitosis and cytokinesis. Cytokinesis is division of the cytoplasm. Usually the 2 go together. You're going to split the whole thing, but you can have cells that will only divide up the nucleus, and you end up with multiple nuclei in there. So meiosis is your sex cell division. This is going to give non-identical daughter cells that are going to only have half the chromosomes as the parent cell. They have half from one parent, half from the other. What has to take place for this to happen is you have fertilization, where you have your two gametes fused together and return the chromosomes back to the normal number in there. So that would be what we think of in the human as the sperm and egg coming together. And I think that is a good place to stop today.